Presentation mode. Is that right? Does everyone see full? I'm seeing a couple thumbs. Okay.
We've got so many pre-installed. I don't even know where to start. Yeah, your slides are big.
Good.
So, yeah.
So, well, it's a pleasure to be here. Thank you, Steve. It was a pleasure meeting you, and I was fortunate to attend the NASPCC. It was the first time I had been in attendance. I'm now part of their board, and I love really being active in this area. Not only because I'm developing therapies, hopefully, that will one day change the clinical practice for prostate cancer and other diseases where we desperately need therapies, but the things that I do in the laboratory as a research investigator actually get translated to the clinic. So I'm fortunate to be part of that process. I'm an assistant professor in the departments of hematology and hematopoietic cell transplantation and immuno-oncology at City of Hope. It's a comprehensive cancer center. Many of you may have heard of it. We're in Duarte, which is outside of Los Angeles, California. Have all of you heard of City of Hope?
No,
I don't think so. I don't
Well,
think there's been a whole lot. I had to Google you on you guys. Kansas City.
All right.
Yeah, you are going to get a chance to introduce City of Hope to Kansas City right here.
Well, I'm going to get in trouble because you're recording this, but I'm going to say that I was born in London, England, but we moved to Los Angeles when I was 3 years old. So I've been here for many, many years. And I went to undergrad at UC Santa Barbara. I got my bachelor's in microbiology. I then worked at a company called Amgen, which many of you have heard of. It's in Thousand Oaks in California, just up the street. I went to UCLA and got my PhD. And when I finished around 2010, I was deciding where to go next. Well, the usual process is to become a research fellow or a postdoctoral fellow and do some research in training before you ultimately go and do whatever you're going to do, either go to industry and be a scientist, or stay in academia and be a research investigator, a principal investigator, or a professor. And I was searching for places and I found City of Hope, and I had never heard of them either, but I was 20 miles away. I mean, arguably I should have known exactly what City of Hope was, but when I found out, I was rather ashamed that I had never heard of City of Hope. Actually, it has a long history. It started over 100 years ago as a tuberculosis treatment center, sanatorium, I think. And when TB sort of got handled in the 50s and 60s, I guess, we had to redefine ourselves at City of Hope, and we became a cancer treatment center, and then subsequently expanded to become a research hospital where we had laboratories and did laboratory research. We have a clinical hospital, and we actually train and give PhDs to budding scientists. We have an active academic program to give PhDs in biological sciences and translational medicine now. We enroll about 15 to 20 students every year. And because of that status, we became one of the 50 or 55 comprehensive NCI designated, National Cancer Institute designated comprehensive cancer centers in the nation, of the likes of MD Anderson and UCLA and USC and Harvard, Dana-Farber, and other places that are part of that club. So we should be known, and especially 20 miles away, I should have known exactly what City of Hope was. But I came here, and really City of Hope has expertise in just a few things. We prioritize treatment of cancer and research underlying the diagnosis and new therapies that may change the treatment of cancer. Various cancers. We actually started 40 plus years ago in doing bone marrow transplants. And at the time when people didn't do what's called mismatched transplants or allogeneic transplants, where patient A gives bone marrow to patient B, which was scary in the 70s and 80s. Now we do that all the time. I think we just reached over 40,000 bone marrow transplants, which we're probably up there, if not the leading bone marrow transplant center in the nation. And we also investigate and find treatments for other autoimmune and inflammatory diseases, including type 1 and type 2 diabetes and other autoimmune diseases. We have a COVID vaccine that was developed here at City of Hope, was just licensed to a company, and I don't know if it'll matter now, but we do that type of research to treat all types of inflammatory diseases. That's all I can say about City of Hope, really. And I came here in 2010, and I started researching tumor immunology and immunotherapy with the idea that you can redirect the immune system to see cancer. Arguably, and I'll just give you this, what I believe to be a fact, but it's a theory to date, is that every one of us get cancer. We probably get cancer before we're even born. And we probably have thousands of cancers trying to get at us until we pass away. And it's the one or two cancers that evade the immune system, the initial recognition that it's a foreign substance, that it shouldn't be growing in your body, that it's eradicated and you never deal with it. You never become symptomatic, you don't even know. That's probably the reason why people don't want you doing PET and MRI scans every year from childhood, because you'd probably identify a bunch of cancers that you may not see the next year. With that said, we know this concept called immune editing, which is the immune system has one chance at seeing this cancer at its very early inception. When a cell goes haywire, it's transformed, it's no longer under the control mechanisms that regulate how many cell divisions it sees, how many mutations in the DNA it can bear before it becomes erratic in growth. And you have that one shot. And when you don't, the immune system has been edited out of the cancer life cycle, and that cancer can grow. And then, you know, depending on what's called immunogenicity of the cancer can grow even in the presence of your immune system recognizing that it's foreign. It's just not mounting an adequate immune response to overcome the tumor growth. But because of the things we've learned on how the immune system responds to viruses and cancer, we reason that the immune system should be able to be redirected or re-engineered to identify the cancer in a very potent manner so that you can overcome the cancer even in the advanced stage. That's what my hypothesis is, and that's what my laboratory has been focusing on over the last several years, particularly in prostate cancer, although we deal with many solid tumors, and I'll talk about that. So that was my big introduction. This disclosure slide has nothing to do with anything that I'm going to talk about today, but it is just to say that whether we develop therapies in my laboratory and keep it at City of Hope, or we license them to companies that can expand on what we're doing and expedite the process to human and the commercialization process. I'm also involved with companies that are developing other cell therapies. I help them develop it with a grand scheme of trying to get things to patients in the fastest time possible, and to advance therapies we believe actually will change the course of the cancer journey. So the pillars of cancer therapy were dominated up until about 10 to 20 years ago by the following, which is surgery. If it's an early stage disease, surgery can be absolutely curative. Usually surgery is intertwined with radiation therapy and or chemotherapy or other targeted agents to provide durable curative responses. And over the last 20 years, we've really reasoned that the immune system and immune-based therapies or immunotherapies, especially in the advanced stage, can offer a lot of benefit to patients, and often has been curative, and I'll talk about that, and sort of gives hope and highlights the potential of immunotherapy for cancer patients. And there have been multiple cancer immunotherapy modalities. These are different ways that you can teach the immune system to see and kill cancer. It arguably started with antibody immunotherapy. If you,
I'm gonna mute him. Hang
Hi,
on.
Scott.
Okay, I think I got him.
that's okay. My email goes off and you're going to hear loud noises too. So I understand.
I was wondering whose beep it was. I think that is. Yeah.
The world of virtual, the virtual.
Yeah.
So
it's a beautiful thing though.
it really is. So if you know someone who has breast cancer, you've heard of the term HER2. And HER2 is an oncogene or a gene that drives some HER2 positive breast cancer patients. It's in about 15 to 20% of breast cancer patients. And they've developed therapies, antibodies that target the HER2 protein, which is on the surface of the tumor cell. You can see that here in orange. And if you're HER2 positive, then you have HER2 protein sitting on the surface of the cell, all over the cell. And so pretty smart people generated these monoclonal antibodies that bind, these are Y-shaped antibodies that bind to the HER2 protein on the surface. And they actually do multiple things. One major thing that it does is it blocks the ability of HER2 to actually signal to whatever it is meant to signal to. And because HER2 is a driver of cancer, if you block its ability to signal, then you block the HER2 positive cancer. And that's really effective. This actual example is rituximab. It's a CD20 antibody. So on non-Hodgkin's lymphoma tumor cells, there's CD20 on the surface, and that antibody blocks CD20 signaling. The other thing that these antibodies do is it helps the immune system recognize the antibody. It actually recognizes the bottom of this Y shape of the antibody. And when it does that, these what are called cytotoxic killer cells can recognize that and actually help in killing the cancer cell. It does that through similar mechanisms as you would imagine if a virus was inside of a cell. And it can kill and it can cause cell lysis. It actually, these dark orange bubbles are actually enzymes that pop holes in the surface of that tumor cell and compromise the integrity of the tumor cell, so much so that it has to die. It's forced to die. And that's really effective. That was the earliest form of immunotherapy. It's sort of we call it an indirect immunotherapy. And then there are things called cancer vaccines. You will have heard of this because Provenge or Sipuleucel T is a prostate cancer vaccine that has been FDA approved. And this is so many of you know this, I would imagine. It's part of the therapeutic arsenal for advanced prostate cancer. And this is a concept where you take immune cells out, they're specifically called dendritic cells or antigen presenting cells, and you load those dendritic cells in a culture dish with proteins that mimic the cancer cell. And what that cell does is it gobbles it up and then presents it on the surface so that it can teach the adaptive immune system what it has seen. It presents the antigen. That's why it's called an antigen presenting cell. And it presents the antigen to these T cells. There are CD4 T cells, CD8 T cells. Those T cells become activated and over the course of a couple of weeks can then specifically see the prostate cancer. It's the same thing as if you cut your skin and a bunch of pathogens land on your skin, which it does, and invade your blood vessel that you've disrupted by cutting yourself. You would succumb to an infection. But what happens is you have these antigen presenting cells that go through the bloodstream, exit, find that pathogen, gobble it up, then travel back through your vasculature to your lymphatic, the closest lymphatic, which is maybe your armpit or axial lymph nodes. And then it teaches the T cells what it sees. And then those T cells go around the body over the next couple of weeks after they're trained and find exactly what your infected cells, specific to that virus. It's the same concept as a typical vaccine, actually, the COVID vaccine, for instance, where they're injecting you with spike mRNA. The mRNA gets into these cells, it produces protein, it's gobbled up by these antigen presenting cells, and then teaches the T cell what to look for, which is spike in this case. Does that make sense? That's pretty crude way
Come
of.
on, guys. I thought when I heard this the first time, I thought, I think I understand it enough.
You can ask questions at any time.
Yeah.
He's talking enough English that I can
Yeah,
get it.
yeah, yeah.
All right.
Yeah, you're brilliant at presenting this incredibly complex stuff.
Okay, because look, on slide 46, it's going to get real complicated, and then it's we're on slide 3, okay, and then on slide 52, there's a pop quiz. No, I'm kidding. I tell it so well because
so the lymph nodes, I didn't know what they were even for. I had heard the term, I knew we had them, but that was the first time anybody ever explained to me what a lymph node was for.
Oh, so you can have me back and I can teach you what a thymus is and what a bone, what happens in the bone marrow. It's all to train your immune system. It's all to get ready for the immune system not to see your pancreas, but to see the virus that you have not seen before, or the cancer that just started to emerge in the body. So there's no, the idea is that you're not creating autoimmunity, but you're creating antiviral or anti-cancer immunity. Okay, so then, and I only know this because, and I'll get to you in a second, but I only know this because I teach my parents this every week. Every week they want to know what I do, and every week I got to go through the same thing. So I can't see your name on here for whatever reason, but someone raised their hand if they want to ask a question.
Yes.
Yes. Hi.
Yeah, I'm a teacher too. I had to explain to my mom what I did all day long.
Yeah.
Teaching. Now, the question I had is, wasn't the mRNA type therapy that we use for COVID first developed for things like prostate cancer?
Yeah, I mean, it was tested for a number of other diseases, as then you probably well know. It was never really tested for antiviral vaccination. It was tested in other diseases. This happens a lot. Like, you develop a therapy for one thing and it doesn't really work out, or there's little need. And then something comes up where you say, oh, why can't we apply what we did here and redirect it for this? And then once you show that it works, now there's all this promise around going back. So now Mrna and Pfizer has been doing it, but Moderna is now like, okay, we're going to create mRNA therapies for cancer. They probably did it in the beginning, but it wasn't really effective. But now we've learned so much about how to make it effective for viruses that it stimulates new ideas. It's exactly what Provenge was. When Provenge was FDA approved in 2010, it's not the best therapy, okay? But the idea that it prolongs survival and stimulates an immune response in patients is mind-blowing for a patient who has prostate cancer. Prior to 10 years ago, 15 years ago, nobody thought that the immune system could be trained to see prostate cancer. And now we know that's a possibility. So now we're all moving forward in the same direction to figure out how best to train the immune system to cure, to get rid of prostate cancer, not just prolong survival, but actually just eradicate it in a patient. And that's our goal.
Oh, so are you specifically working with mRNA?
No.
Okay. Then that's all I have.
But I think there's a lot of utility there, and I'm happy people are working on it.
Thank you.
Not to say I won't, but I don't. And so the third type of immunotherapy is called bispecific T-cell engagers. You may have heard of it also because there's an active clinical development strategy around bispecific T-cell engagers for prostate cancer, targeting PSMA. which maybe all of you have heard of because of the PSMA imaging opportunities there are to identifying prostate cancer around the body that may not be detected by other conventional scans. But bispecific T-cell engagers are really kind of cool. They're much more active of an immunotherapy strategy than I just told you in the prior two examples. This is really cool, right? So when a T-cell, which I just told you, could go and find crap, right? Find tumor cells and virally infected cell. When it does that through a protein that it expresses on the surface called TCR. And it goes after tumor cells that express what's called MHC. MHC is really cool. So your immune system goes around. So how would you identify a virus in general? A virus sits inside the cell. It doesn't survive very long outside of the cell. So once it's inside the cell, how does your immune system see what's going on in a neighboring cell, in another cell? It does so because every cell in your body has to present the goods that are going on inside of them. They do that through MHC. They present little bits of peptides, little short proteins that encompass every protein that's inside that cell. It's how your immune system would identify a cancer cell, and it's how it does identify viruses. And it does so through presenting peptides through MHC, which is recognized by a T cell through its TCR. But in patients, this system that should be so effective is not effective. It's defective. And there's many mechanisms. I won't go into why exactly that is, but suffice it to say, it doesn't work very well. Bispecific T-cell engagers really just force a T cell and a tumor cell to be in close proximity. It does that through two antibodies that are linked together. One of the antibodies binds the T cell and the other antibody binds the tumor cell, as depicted here. And when it does that, it brings the T cell in such close proximity. Sorry. It does so well to bring a T cell in close proximity and stimulates a T cell just a little bit that it can activate the T cell to kill that tumor cell. And we have FDA approved therapies now. One is called blinatumomab. They all have weird names. Blinatumomab. It's also called Blincyto. And Amgen developed this to target acute lymphocytic leukemia. And it does so because one part of the antibody targets CD3 on the T cell, the other side of the antibody targets CD19, which is a surface marker that we talked about on the last slide that is showering the tumor cell. That's all over the surface of the tumor cell. Very effective therapies. People are developing this all over the place, including Amgen has a PSMA targeted bispecific T-cell engager that is showing pretty decent activity in prostate cancer. And then really arguably the most effective and widely vetted immunotherapy approach to date, prior to CAR T cells, which I'll tell you about in a second, is checkpoint blocking therapies. And now, okay, so I'm gonna go a little backwards and talk about the same example I gave you where you cut your skin and you have a bunch of nasty things that landed in the cut and you're succumbing to a bunch of infections, except for your immune system is recognizing it and getting rid of it. When it does recognize it and gets rid of it, it only takes one T cell in all of your body where there are trillions of cells, one T cell to recognize that virus or that bacteria or whatever. And it can ramp up from one cell to millions of cells. And that's how it does it. It literally overpowers the pathogen in number, and it has the potential to kill it. And so it does that to millions. If there wasn't a resolution of that immune response, you would succumb, you wouldn't succumb, but you would be inflicted with a chronic inflammation, because your immune system is ramped up and nothing has shut it down. Evolutionarily, you have a contraction phase of that immune response. So it comes up, it defeats the virus, and then it has to resolve. That resolving process is we know exactly how that is mechanistically in the body. And cancers have hijacked that to literally shut off the immune system inside the cancer. It uses the same mechanisms that we use in evolution to resolve an immune response. When the immune response is completed, they continually shut it off. And so pretty smart scientists, actually in 2018, 2 scientists earned the Nobel Prize for this understanding that cancers can use the same mechanisms your normal immune system uses to be at a low, and shuts off the immune response. When you use these antibodies that block these pathways called checkpoint pathways, and you block them, you allow the immune system to go back to what it was inside the cancer at the height of recognizing a foreign thing like a cancer, and the immune system works again. We have multiple approvals by the FDA of these therapies, and it was led really by metastatic melanoma, where they were first indicated, and now it's used to treat renal cell cancer and lymphomas and ovarian cancers, some breast cancers, and other diseases. Unfortunately, it works really poorly for prostate cancer, and I'll go into why that is. Actually, I'll go into why that is right now, because the immune system in prostate cancer is rather silent. There's not a big recognition by the immune system that there's a prostate cancer growing inside of us. But in melanoma, there is. Your immune system is desperate to target the melanoma. The cancer has shut down the immune response. You provide these checkpoint blocking antibodies, the immune system can go back up, recognize the cancer, and you get an anti-tumor response. It's very effective in a subset of patients. In fact, in 10 to 20% of patients that are on these therapies who have melanoma, you not only get an initial response, but you're literally cured of your disease. You go from heavily metastatic disease burden to cancer-free, and the responses can be forever, which is something that we've never really seen before with other targeted agents. And you don't have to continue taking the therapy. You can actually take it several times. Once the immune system is ramped up and ready to go, you've trained the immune system. It's actually a living drug. It's not a drug you have to continue giving. You've literally changed the blueprint of the immune system, and now it recognizes the cancer, and there's no way to stop that, thankfully. Okay, so those are.
We have to keep getting mRNA COVID shots if the immune system learns how to use it.
This is a prostate cancer discussion. I refuse to talk about that. Yeah, it's a good question. I mean, some viruses mutate. Some viruses are really responsive to vaccines durably, like, for instance, polio vaccine and measles, mumps, rubella vaccines and others. And some you constantly need to take it, like influenza vaccine. You're taking it every year. It's because the virus changes, and we don't understand how to predict those changes and how those changes occur in a meaningful, we can't model that appropriately. So because they change, your immune system is no longer effective against it. And actually, we're realizing that could hold true and does hold true in cancer immunotherapy. So there's still a lot to learn.
All right, I'll get focused on prostate. Sorry.
Yeah, it's okay. We can talk about everything. I'm excited to talk about everything. But there are reasons that I know of, and some things that it's inexplicable, and I won't be able to give you an answer to. And I'm happy to say that. I'm not ashamed of not knowing everything. So then 20 or so years ago, because. And now you're experts, right? So TCR on the T cell sees MHC on the virus infected cell or the cancer cell. They talk to each other, and then the T cell can kill the bad cell. And I also told you that MHC and the whole machinery in processing peptides and proteins that are inside of the virally infected cell or the cancer cell, that becomes defective. It's not always working properly. And that's why in large part, also the COVID vaccine and other vaccines don't work. Because of that, people thought, well, T cells are really effective, right? If you train a T cell to see a cancer cell, do you really need it to be presented on MHC for the T cell to recognize? And the answer is no. If you can engineer, and we do now, T cells to express a different protein on the surface that recognizes the cancer cell in a different way, we call it MHC independent way. Can that T cell still become cytotoxic and active and kill that tumor cell? And you never needed it to see MHC? And the answer is yes. We call it chimeric antigen receptor technology. It basically looks like this. You have an antibody like protein here on the surface. This is the surface of a T cell. You have a transmembrane domain, which goes through the membrane of the T cell. And then you have this intracellular signaling domains, which allow, once this antibody to see the tumor cell, it engages the T cell, activates a bunch of things inside the T cell, which allow that T cell to become active and kill the tumor cell. That's the concept of chimeric antigen receptor T cell therapies. And because the whole immunotherapy field has exploded, as I mentioned, over the last 20 years, you can see here how many individual therapies there are across multiple modalities. You have T cell targeted immunomodulators. At the bottom, you have these bispecific T cell engagers I talked about, cancer vaccines, cell therapy, which is where I live, which is actually leading the pack because there's so much promise. All of these are different indications, different diseases, different therapies from the preclinical stage all the way to commercialization. The field is exploding because we understand how promising immunotherapy might be in treating cancer. And at the top here is these are the targets which we're going after. And CD19, as I explained earlier, is on leukemias and lymphomas and is the subject and the target of multiple therapies, from bispecific T cell engagers to chimeric antigen receptor T cell therapies. And the way that we do this is quite simplified here. At City of Hope, we do this a lot, and a bunch of places around the nation and the globe do this, is autologous CAR T cell therapy, which is patient A, we harvest blood from in what's called an apheresis. It's a large amount of white blood cells. We bring it to the lab. We enrich those T cells from the blood, because the blood actually houses about 20 to 30% of the white blood cells in the blood are T cells. So we take those T cells out, we engineer them to express the chimeric antigen receptor protein on the surface of that T cell. And we grow them up from hundreds of million cells to billions of cells. We freeze them back. We do all sorts of quality control to make sure they're sterile and active and ready. And then they get infused back into that same patient. That's called autologous. Patient A provides the therapeutic product for themselves. That makes sense? Okay, so we've done this for. I think if you just answer yes or nod, then you've passed the quiz.
Okay, all right.
Yes.
So just to give you an where we are in terms of how many patients we've treated across a number of different diseases. This is abbreviated, but I'll explain it to you. So this is non-Hodgkin's lymphoma. This is acute lymphocytic leukemia. This is acute myelogenous leukemia, AML. This is multiple myeloma, which is, you would know it as myeloma. This is GBM, or glioblastoma, a very deadly brain tumor. This is BBM, which is, well, we call it breast to brain metastasis, but remember I told you that HER2 positivity is in about 15 to 30% of breast cancer patients. Well, unfortunately, because therapies are so effective in treating breast cancer patients who have metastasis not in the brain, almost 50% of those patients ultimately develop brain metastatic disease, because the cancer cells are so sneaky. They get through the blood-brain barrier where our drugs can't easily get to, and they set up shop in the brain. It's very, it carries a very poor prognosis, as you can imagine, and is in desperate need of new therapies. PSCA for MCRPC, or as you know, and I'll talk about that, and other diseases. And we've treated about 50 to 70 patients a year over the last 4 years, and we're ramping that up a lot. So at City of Hope, we're not only trying to develop new therapies, but we're trying to apply them across a number of diseases to try and impact the lives of anyone who has cancer. But also it's a strategic plan because we actually learn from lymphoma trials, and they inform on prostate cancer trials, two very different diseases, but because the underlying therapy is similar, we can cross-fertilize and learn across a number of diseases. So as I mentioned, CAR T cells have been very effective for treating hematologic malignancies. They're actually FDA approved. There's 5 FDA approvals now to treat leukemias, lymphomas, and just recently, about a year ago, for patients with multiple myeloma. It's been very effective, the CAR T cell field, for liquid cancers. And we're really trying to apply it to solid tumors now, and I'll talk about that. Here's an example of a patient at City of Hope with a B-cell lymphoma, and you can see the disease all over here, and treated with CAR T cells targeting CD19, and you can see within 1 month, the patient is disease-free. We've actually seen this in a high number of patients treated with leukemia and lymphoma. Overall, about 90% of patients who have leukemia who are treated with CD19 CARs go into complete remission, and over 40 to 50% of those stay in durable remission, which is pretty striking. These are patients that have seen a ton of other therapies prior to coming on these trials. So now the effort is into moving it up front in frontline therapy for patients who are newly diagnosed. And so that's where we are with the heme malignancy. And you can see this in a different chart here. On the top are all the targets for hematologic malignancies across a number of diseases, leukemias, lymphomas of different sorts and flavors, and all the targets you go after. CD19, as I mentioned, has the highest number of clinical trials that are going on, but you can see all the other ones are following up. And now we have this FDA approval of BCMA-directed T cells for multiple myeloma. And on the bottom, you can see—
Can I ask a question before we?
Of course.
Back to the prior cell, since you opened up about other cancers. I'm
Yeah.
a 7-year survivor of prostate cancer, but recently been diagnosed with metastatic stage 4 colon cancer. In all of the cancers you mentioned, is there anything immunotherapy on the horizon?
For colon cancer?
Yes, sir.
There's a lot of work being done in colon cancer, actually. I would talk to your oncologist and discuss options, but there are quite a significant amount of options. I don't know your exact disease and where you are in your treatment, so I won't speak on what's available to you specifically, but I would argue there are several options you could explore in the realm of immunotherapies.
All right. Much appreciated then. Any specific research labs like yours?
Well, the NIH actually has a couple of labs who are active in developing immunotherapies for colon cancer. I'd have to do a little bit of specific research, and I'm happy to, if Steve provides you with my email, I'm happy to do a little more digging for you.
Okay, I'll deal with that then with Steve. That would be great.
I'll send you guys both an email. George is one of our favorite guys. He's been helping us for so many years. If there's anything you could do to help George, it would be incredibly appreciated.
I'm happy to, and/or put you in touch with people that would.
Awesome. I'll send you guys both an email after this.
Okay.
Thanks, Steve.
So on the bottom here is a graph of targets that people are looking at for solid tumors, and they span all different cancers. In fact, George, CEA here is a target that people are actively looking at for colon cancer, for instance, and is also a target in neuroendocrine or small cell prostate cancer that we're looking at. In terms of prostate cancer, though, there are two heavily active targets, PSCA or prostate stem cell antigen, and PSMA, prostate specific membrane antigen. Many of you may have heard of that because lutetium PSMA is a very active agent being tested and near to approval, if not already approved, for prostate cancer. And not to say this always happens, but we published this study several years ago now in a case report of a patient with recurrent glioblastoma, again, a very deadly brain tumor, with CAR T-cell therapy, and put him into a nearly 8-month complete remission, which is unheard of for that disease. Ultimately, he recurred and he passed away 3 years later. He was given, I think, several weeks to live prior to therapy. So while it didn't cure him, it certainly provided him additional years of life, which was amazing. We published that. It was amazing enough to be published in the New England Journal of Medicine. And we continue to advance efforts in that disease. I work with several people at City of Hope, including Tanya Dorf, who's a fantastic clinical oncologist, medical oncologist here at City of Hope, treating primarily prostate cancer patients, as well as folks at the University of Pennsylvania. And we published a review late last year in basically reviewing all the active therapies that folks are looking at, including BiTEs or bispecific T-cell engagers, which you now are experts in, and CAR T-cells also, which you are experts in, and all the different clinical trials that are ongoing now, active in trying to treat patients with prostate cancer with immunotherapies. And you can look at that review, and I'm happy to forward it to Steve as well. It's a great read, not because I was part of the writing, but it actually gives a more recent update on the types of therapies, and is a good read in terms of reviewing the types of immunotherapies that might be active for prostate cancer patients.
Yeah, I want to ask a question. So if you go back to that last slide, so if I'm understanding this right, these are all active clinical trials for using immunotherapy to treat prostate cancer, right?
That's
and
right.
So in all likelihood, there's probably some guys on here that might qualify for one of those trials, but now we need to figure out how we would figure out who.
That's right. So if you go to this,
who.
Yeah, so I should really do a training on how to identify clinical trials, because that's just not done, which is a shame. But this is the NCT number. This is the clinical trial number,
okay,
that you can type into Google, and it will tell you all the eligibility criteria. Not that you'll understand it all. I don't understand it all, but it will tell you the contact or the coordinator that's heading up that trial. You can call them directly, and you can go through the whole process of understanding whether you're eligible for that trial. But I would talk to your clinical team as well to figure out what's probably the best therapy for you to be on. One of the largest considerations is what the target is and whether you express that target if you have prostate cancer.
Yeah, so if we have a guy on here whose PSMA scan is lighting up, one of those PSMAs is likely a good idea, maybe, right?
That's right.
Yeah, and I know there's somebody on here with a PSMA lighting up.
Most
So
of
there
you,
you go.
most
Yeah,
of you on here with prostate cancer will have PSMA
Yeah,
and PSC.
thank you.
So just going back, so again, at City of Hope, we really treat a large spectrum of cancers. In the hematologic malignancies or liquid cancers, in the middle, which is where I sit, and I develop therapies for prostate cancer. Largely, my big focus is prostate cancer, but I also have active clinical trials in breast cancer and recent FDA clearance to do a trial in ovarian cancer, which will treat our very first patient in the next month or two. And I've been part of programs that have developed pancreatic cancer immunotherapies and liver cancer. But we also treat actively brain cancer. Many years ago, and this was actually 2014, we looked at many targets for prostate cancer. We narrowed in on prostate stem cell antigen. In addition to the idea that the fact that it's a great target in prostate cancer, it's not only a prognostic indicator of prostate cancer, but high PSCA is found in more advanced disease. The expression is found high in primary prostate cancers, but also even higher in the metastatic tumors. And there's limited expression of it in normal tissue. There's some expression in the bladder. There's been some observed expression in the stomach, but largely very minimal expression in normal tissue, which makes it a good candidate for CAR T-cell therapies. Not to mention, we have an active collaboration with folks at UCLA, which are leading a big charge in developing prostate cancer therapies. And they actually identified PSCA first in 1998, and it's been studied ever since. Well, we went in and said, okay, that's a good target. And we developed a PSCA-directed CAR T-cell strategy. And this is just a mouse study because this all starts of what happens in the dish and then what happens in a mouse. And then we can start a trial. This was published now 3 years ago, but I just thought it would be cool for you guys to see what happens in the laboratory, which is that we take a mouse and we inject that mouse, in this case, with a prostate cancer. This was actually a prostate cancer that was harvested from a patient with bone metastatic prostate cancer at UCLA. And they continued to grow that tumor in the laboratory, and we use that to study it because it's really closely relevant to what's happening in patients, we think. And we engraft that into the mouse. And we actually label that tumor cell with what's called firefly luciferase, which is the enzyme that fireflies use to light up. And we use that enzyme to light the tumor up. And you can actually see that non-invasively over time. You see in this mouse at day 13 post-engraftment, it's in the bone here. And it grows and grows and grows and grows and grows and grows until it takes over the whole body and the mouse dies of prostate cancer. And when you treat it with a CAR T-cell, that tumor goes away. In some of these CARs, which we build, because the CAR is a synthetic receptor, we're actually modifying multiple things, proteins, and fusing them together to recreate a receptor on the surface of the T-cell. You can imagine that each one of those domains, each one of those proteins that we're putting in one long tandem sequence could be modified or optimized to further improve the therapy. This is just an example of 2 PSCA CAR T-cell therapies. I won't go into detail on what they were, but they both targeted PSCA. Just the receptor changed downstream of the PSCA targeting antibody. And you can see that both eradicated disease, but by day 34 in the animal, this CAR couldn't seem to keep up with the tumor, and the tumor grew back and the mouse died. And in this CAR, it took a little longer for the tumor to go away, but when it went away, it was gone in 90% of the animals forever. Those mice lived happily ever after with no prostate cancer. We looked at other things like PSA in the blood, and the PSA was not detected in those animals. We looked at the CAR T cells in the blood because it's a living drug. It should be sitting around in circulation in the mouse. And those CAR T cells lasted for longer than the other CAR T cells. You could see the blue or cyan versus the gray. And that probably is the reason why those 2 different CAR constructs acted very differently. One just didn't last that long in the body, and the tumors were able to recur. The other one lasted long enough to eradicate the tumor. A lot of different studies went into this package. We went to the FDA, and the FDA allowed us to start a trial at the end of 2019, and we've been in trial ever since. It's pretty slow in phase 1. We've treated 12 patients over the last 2 years. With COVID especially, it put limits on patients being treated, what we could do. But the trial is as follows. We have prostate cancer patients, metastatic castration-resistant prostate cancer. They're consented onto the trial. They're screened for PSCA expression. Remember, that target has to be on the prostate cancer for us to want to develop a CAR T-cell targeting PSCA. Once they're screened and positive in a biopsy, they're apheresed. We manufacture the T-cell here at City of Hope. We produce it. We do a baseline CT scan. We take blood. We give what's called a lymphodepleting chemotherapy. It's cyclophosphamide. You may have heard of that. And I'm going to give you preclinical data in a second as to why we're using that in prostate cancer, where otherwise it's not really used. And then we infuse the CAR T-cell, and we track that patient over 28 days. After the 28 days, they do another CT scan and a bone scan and a biopsy to understand if the therapy is working. Does that make sense? Yep.
Brilliant.
Well, the question I had was if the only imaging you're using are a MRI or CT scan, and you're not using anything to specifically go after the PSCA?
Yeah, very good question. So when we started the trial, there's always funding issues. I mean, so to make this therapy, just to give you an example, and maybe we can, redact this in the recording. It's about $150,000 to develop the therapy for a patient. So I work really hard to find funding to do that so it doesn't cost anything for the patient. But it's a big expense. So anything else you add on to that, it becomes even more expensive, obviously. But what we tried to understand was get the best idea. So we did bone scans, CT, and MRI, and try and get the best idea of the tumor. Now, City of Hope is actively developing a PET scan for PSCA directly, and our future trials are actually incorporating PSMA PET imaging because it's working so well at identifying cancers that may not be seen by MRI or bone scans. So great question, and we're trying to incorporate the most advanced imaging modalities for our trials.
I've had several PSMAs, but I never heard about having a PSCA scan, and I didn't know if they'd put them together and just go after them both.
It's not approved yet, and there's early research in it. It's just not as advanced as PSMA as an imaging tool yet.
Are they overlapping in what they can detect, and the PSMA is just better?
They have some overlap. I wouldn't say it's better, but they have some overlap. There are some differences, though. So it's worth developing a PSCA imaging modality.
Thank you.
So how do we build preclinical models, these animal models, to best inform on therapeutic outcomes for patients? And this is one of the things that I work on so intimately in the lab. And just to go back to why we're adding cyclophosphamide and how we developed this model to understand the therapy, we actually built a fully clinically relevant mouse model in the lab that we could actually test safety as well as efficacy of this PSCA CAR T cell. This was actually first in field. No one had ever really done this before. And we published this just a couple of months ago. And I won't belabor the details of this, but suffice it to say, the mouse expresses low levels of PSCA in tissues that we know are expressed in patients and humans. And there's a full immune system in this mouse, unlike the prior study that I showed you where the mouse has no immune system. So we can inject human tumor cells, we can inject human CAR T cells, and the mouse doesn't reject the human stuff because it's foreign. If the immune system is intact in the mouse, it will actually reject human tumors and other cells. Does that make sense? So usually people test human-related therapies in a mouse that has no immune system, so it can't reject anything. But we said, well, your immune system is really critical to this therapy. We have to allow the immune system to work. So in this model, we did. And what was really striking is when we gave the PSCA CAR T cells to these mice, we didn't see any therapy. It cured the other mice. Like, what's going on here? You put an immune system in the mouse and all of a sudden it the CAR T cells can't see the cancer. But when we precondition those mice with cyclophosphamide, here it's labeled CPA, which actually has its own therapeutic response. You see the tumors growing slowly here, but then they grow out and all the mice die of cancer. When we follow that cyclophosphamide now with CAR T cells, we cured 50% of the animals. So that's why we built cyclophosphamide as a lymphodepleting agent into our clinical trial. Cyclophosphamide is used in every clinical study with CAR T cells and hematologic malignancies like leukemia and lymphoma, but it has no business being in prostate cancer, really. It's really a standard of care in liquid cancers. We had to prove that it would have any effect here, and it actually did. When we went back and understood what was happening, this is a tumor histology. I don't know if you've looked at histology before, but it's basically a two-dimensional slice of a tumor. And if I zoomed in, you could actually see individual tumor cells, but these are probably 50,000 tumor cells sitting here. Oops. And when you look at the tumor area, and this is marked here, where the tumor stops and the normal tissue starts. And if you stain for T cells, you can see all the T cells sitting in the normal area. No T cells are coming inside the tumor. It's called T cell exclusion. It's one of the reasons why prostate cancers are silent in their immune system. The T cells want to get in, but they're being prevented from getting in. And when we gave the CAR T cells, it was the same thing. All those T cells are sitting outside the tumor. When we gave cyclophosphamide, you started to see these brown dots and T cells sitting inside the tumor. And when you gave the CAR T cells, they were really inside the tumor. We did this by imaging, actually. We remember firefly luciferase, you can put inside a cell and you can image that cell non-invasively. Well, we put that firefly luciferase inside the CAR T cell this time, not the tumor cell. And you can see the T cells get into the tumor and start growing and growing and growing in numbers inside the tumor. Versus without cyclophosphamide, they get there, but they're really sitting outside. You can actually see a donut shape of these T cells sitting outside the tumor. Do you see that? And if you quantify those T cells, this is a log growth. You can see these T cells are like 10 times, even 100 times more frequent inside the tumor with cyclophosphamide preconditioning than without. And when we looked at the transcript levels and protein and RNA and other things inside the tumor, you can actually see really dynamic shifts of the immune system inside the tumor. And we knew that there was some inflammatory thing happening inside the tumor and causing it to go away in 50% of the animals treated. And so we published this nice paper and basically proved that we think cyclophosphamide preconditioning is important, and that's why we built it into our trial. And it really brings up the concept that some tumors are immunologically warm. That means there's an immune response happening, like melanoma. And there are a ton of tumors that are immunologically cold, where there's just no immune system happening inside the tumor. And that, unfortunately, is prostate cancer. And that is fortunately why we're developing CAR T cells where we're bringing the immune system into the tumor. We don't care what's actually pre-existing there. But it is the reason why immune checkpoint blockade doesn't really work in prostate cancer, although in some patients it does. And I would argue that you need to explore your genetics and what's happening in your specific tumor, because you may be eligible for a checkpoint therapy that might be really effective in you, and it may not be effective in the royal we as prostate cancer group. Just things you should talk about with your clinical team. But it also has highlighted things like tumor heterogeneity, which is not all tumor cells are alike. Even if they're sitting together or there's a bone metastasis in your leg and in your arm, they're two different tumors really. And it's really tough to target both of those with one type of therapy. And also the prostate cancers are immune suppressive. And I just showed you an image of that. The T cells are sitting outside the tumor. The tumor is literally suppressing the immune system from coming inside the tumor. And we're getting around that in many ways. So it's brought up what is the most rational combination of therapies that one should use to improve on the immunotherapy response. So remember I talked to you about this prostate cancer model. We built it. We tested our Psca CAR T cells. They didn't work at all. And then we gave cyclophosphamide, and now the CAR T cells cured 50% of the mice. But 50% isn't 100%, and we want to get to 100%. So we actually did genetic studies in the tumors that recurred after Psca CAR T cell therapy and figured out a couple of pathways that were important, that were coming up. They were lighting up in these tumors that were recurring after the therapy. And we designed a therapy directed at one of those pathways. And when we combined it with our CAR T cell, remember, the CAR T cell plus cyclophosphamide, and this is a different study, different mouse study, so you can see how reproducible the data is. 3 of the animals came back in their tumors. 3 of the animals were cured. That's 50% with the CAR plus cyclophosphamide. And then when we gave CAR plus cyclophosphamide plus this new therapy, we actually only had 1 straggler tumor that grew out, even though it was initially cured. And 5 out of 6 of those animals were completely cured. We now bumped the therapeutic response to over 85, 90%. So now 90% of the mice are cured. We think this is an active therapeutic combination, and we're bringing this to trial very soon. I'm going to blow through this, I think, because I'm already over an hour and I don't want to bore people. But we're also investigating other therapies. Oncolytic viruses are really cool. So this is apropos because we're talking about COVID, and oncolytic viruses are really cool. They actually see cancer cells more than they see normal cells. Sometimes they only see cancer cells. They don't see normal cells. So if you infuse it into a patient, it can go seek around and find a cancer cell and replicate in that cancer cell and kill the cancer cell. And it actually causes what's called immunogenic cell death. So in addition to killing the tumor cell, it recruits the immune response to the tumor cell. And with all that, you can actually engineer a virus to express any gene you want. So you can amplify an immune response, or you can improve the ability of it to kill. There are FDA approved oncolytic viruses. Actually, Amgen, again, really is leading the charge. They have what's called a herpes simplex virus that they engineered to be an oncolytic virus, and it's used for the treatment of melanoma patients. But there are challenges as single agent therapies. So what we did was kind of cool. Now that you guys are experts, and I think I have all this data that I could show you, we published recently, but I'm going to show you this illustration because it will tell you exactly what we did, which was really I think, a really kind of groundbreaking thought, a very simple notion. You'll understand it now that you're experts in CAR T cells. Oncolytic viruses, right? They should infect the tumor cell. They should kill that tumor cell, but they don't kill all tumor cells, but they infect some of them. So what if we engineered that virus to express a gene that can redirect the CAR T cell? Well, we can put CD19 into a virus. Virus infects prostate cancer, ovarian cancer, colon cancer, pancreatic cancer, liver cancer, all the cancers you can imagine, but presents CD19 on the surface. And it would literally turn that cancer into a lymphoma. And now you can treat that cancer with the FDA approved CD19 CAR T cell. Whether you have breast cancer, colon, prostate, ovarian, you're going to get that same CD19 CAR T cell to kill it. And we did. We actually showed it did really well in mice. We cured mice with this combination. It didn't matter whether they were PSMA positive, PSCA positive, CEA positive, CD19 positive, because we engineered them to be CD19 positive with the virus. And I'm going to skip past this because it's all this data that you don't need to know, except for this last figure. When we cured the animals with this oncolytic virus coming in, forcing CD19 to be expressed on the tumor cell, and then killing it with the CD19 CAR T cell, in those cured animals, we re-challenged them with tumor cells that never saw the virus and weren't CD19 expressing, and they didn't grow in the animals, suggesting that we not only killed the cancer, but we retrained the immune system to see the cancer, independent of the proteins that it expressed, suggesting that once you cure a patient, the cancer will never have a chance to grow back in that patient, even if it changes. So we've done a lot of studies on that. And so I think
there's a couple of guys that want to get that treatment.
I'll tell you where we are on that actually. So remember I gave you my disclosure slide. So because this is a pretty complicated combination therapy and using an approach that, is FDA approved by a company we don't work with necessarily. And one part of the therapy is the oncolytic virus, which we developed wholly here at City of Hope, but has never been in a human. It's a complicated trial. We actually licensed the technology to a company who's really leading the charge and taking that trial on. We're working very heavily with them to bring it to trial. They're really pushing the timelines, which is awesome. It's driving me nuts, but it's awesome because we'll be in trial way sooner than we would have if it was just coming through from my lab. So that was why I presented the disclosures, because we are working with really amazing partners who will take these things to trial when we can't. So we're learning from phase 1 trials. We have phase 1 trials, as I mentioned, in prostate, brain metastasis, ovarian cancer, and soon to be pancreatic cancer. And we think we've come up with multiple ways to overcome the challenges of solid tumors. And that's the goal of the laboratory. This is like an old picture now. I need to update it, but on the left side here are all the current members in my lab, all the funding sources which allow us to do the research and take things to trial. I'm part of a program that develops immunotherapies across a number of diseases, as I mentioned, so it's really a group effort. And I have the fortune of having a number of amazing collaborators at City of Hope, as well as all over the nation. And as I mentioned, we have a number of industry partners that help us take things to trial. So that's it. And I'm happy to answer any questions.
I just want to thank you so much for not only joining us tonight, but especially for everything that you're doing. I mean, it is incredibly inspiring, and I'm so glad that my guys here got to hear this. So how about we give Saul a hand?
Hey.
Yeah. I want to hit the